40.
Sorry, that was me.
There are 45 representatives from Bayer in the room. So this is a unique program. We've had, like other REU programs, we've had lots of opportunities to play together, to do research together, lots of great things happening in that venue. But also on top of this, we're really pleased. But this is a model REU program, and it's part of a pilot through the ASF of a very successful program. And that was ramped up by Phil Taylor, of having mentors with an industry who can provide insights into career progression for the people standing a few meters away from them. So we're fortunate to have online, actually, the person who came up with this program, Phil Taylor. And Phil, if you'd be willing to just, if you are not in the middle of another Zoom meeting, if you to unmute for a second, love for you to say hello to the gang here. And if you're
yeah, thank you, Guy, and sorry I couldn't be down there with you in person this morning, but thrilled that we've been able to run this program again this summer, and just can't wait to hear what the students have been up to and what they've done. And I'm hearing great things from the mentors in terms of the interactions and the conversations they've been having, and so delighted to be part of the conversation today, and I can't wait to hear. And already, Guy, let's start talking about doing this again next year.
Thank you, Bill. Thank you for being here. Thank you to all the biomentors who are here in person and on Zoom. With that, I'm pleased to introduce yet again the woman behind a lot of the magic here, Patricia Widder.
I just want to add to what Guy has said. This has been one of the most successful REU summers that we've had. I think our students have been the most engaged and the most creative and productive of any summer that we've ever had. So I feel like we've had really good matchups in the labs. You guys have worked really hard. You guys have gotten along well with one another. You've taken advantage of what St. Louis has to offer and what WashU has to offer. We're going to miss you. It's really been a fun summer. Together we have learned about scientific manuscripts. We've learned how to read them. We've learned how to write them. We've learned how to search the literature for what we want to find. We've learned how to create beautiful slides to show to our labs. I know that many of you have given lab presentations to your lab groups of what you've been doing. You've learned how to create a poster, and then you've done so. You've learned how to briefly discuss your work and communicate it to a wide audience. And so I think thinking back to those first couple of days when you were here at the beginning of the summer, I think how far you've come, and we're so proud of all the work that you've done. Thank you to all of the bench mentors, the PIs, the Bayer mentors who have been part of this. We really appreciate that support and guidance that you've given to the students. Anything else we want to say right now? Great, thank you. Okay, so I'm going to make a change here to the posters, and we'll start with Melanie Roberts. I'll let her introduce herself. And let's go. Okay, awesome. Oh, how did I do that? Did it? Oh, maybe I had them out of order. Anyway, there's you. Oh,
okay, cool.
So goes, right?
Awesome, thank you. All right. Hi, everyone. Good morning. My name is Melanie Roberts, and I am a rising junior chemistry major from St. Olaf College. This summer, I've had the privilege of working in Dr. Jessica Wagenseil's vascular mechanics laboratory, and I'm so excited to tell you all about what I've learned and how it connects to the greater context of heart disease research. To start, I'd like to talk about the aorta. The aorta is the main blood vessel leaving your heart, and it takes blood to the rest of the body, so it's really important. Also very dynamic. It's under a lot of stresses due to the pumping of the heart, and it expands and contracts with every single beat. As a result, its structure must reflect this. It's made up of 3 main layers. And I'd like to point out that it has the intima media, which is made up of these dense elastic fibers, which give it this ability to stretch and shrink. And finally, the adventitia on the outside. One way we can study aortas and aortic diseases is looking at transport across the wall. So in this image here, from the lumen to the outside of the adventitia. Previously, our lab has looked at transport in tissue of aneurysm models and mouse models versus healthy tissue, and it saw great changes in the transport across the wall. However, this was very static in very static conditions. Pressure was kept at constant 100 millimeters of mercury, and the stretch was constant at a 1.1 times stretch ratio. But like I mentioned, this is a very dynamic system. So what is actually going on with the transport when we mimic these in vivo conditions? And that's what I had the opportunity to look at this summer. All right. So the main things I looked at were pressure, axial stretch, and the viscosity. And how I did this is I used a myograph system. So I cannulated a ascending aorta onto the myograph and sutured both ends. This made sure that the fluid flowing into the wall was not leaving on each end of the suture, but instead leaving through the wall itself. I then set up a pressure column filled with phosphate buffered saline and introduced a bubble into the tube. The bubble was then placed on I was able to track the distance it moved over time. I could take this dx dt and put it in my equation for fluid. What's important about looking at this equation is it's not only normalized by the diameter of the tubing to see how much fluid is flowing through the wall, but it's also normalized by the geometry of the vessel itself, as seen by the measured diameter of the vessel and the length measured. This can ultimately be translated into hydraulic conductance, which is a measure of the material property of the wall itself as you divide out by pressure. So that's how it was the main experimental setup, but I had these 3 conditions. How was I going to change them? For pressure, I had 3 experimental groups. One was the normal blood pressure, and this was the 120/80. So first, I would set up the vessel, test it at 80 millimeters of mercury, and then move it up to 120. I did the same thing with the hypertensive group, showing 190. And then finally, I had one group that stayed at 100 millimeters of mercury. The other thing I looked at was axial stretch. So I started at a 1.1 times stretch ratio and increased by 0.1 up to a 1.5 times stretch ratio. Finally, I wanted to mimic blood viscosity. So I did one test with our phosphate buffered saline, which is about at 1 centipoise. And then I made an alginate solution, which is basically like seaweed, and it got it to 3.5 centipoise. That's for me to figure out, use the rheometer and make it work. What did we see? I'd like to start talking about pressure. This upper image in red, the elastic lamellae are really close together. They're really struck together. However, when you start to pressurize the vessel, we see a structural change. We see the lamellae begin to spread out, straighten, and stick together. So although we see the structural change, we actually don't see any change in hydraulic conductance. Values are very similar. Then when we look at axial stretch, we see that, again, the mean values didn't change overall, the different axial stretch ratios. And finally, with our viscosity, there's a difference in variance. We saw a greater variance in the phosphate buffered saline rather than the alginate. However, again, the mean values didn't change. We think that this difference in variance is due to some property with how the alginate reacts with the tubing as well as the vessel wall itself. That was a lot of what actually happened, and we didn't see any changes is the main summary, which is great because A, it confirms what we've done previously in our lab, and B, it simplifies all experiments going forward when we look at the mouse ascending aorta and transport. It makes the experiment much easier to do and make the data a lot easier to look at. Other studies have also seen pressure changes not affecting hydraulic conductance in rabbit and rat ascending aorta, but it is the first time it's been done in the mouse ascending aorta. And this is the first connectance study of hydraulic conductance and dependence on axial stretch ever done in any arteries to our knowledge. So all this data can then be used going forward to look at both diseased tissue and regular tissue, as well as looking at how drugs interact with the wall and moving forward into trying to find some solutions to the problem of heart disease in every day. Finally, the smallest thing on my poster is one of the most important, my acknowledgements. I'm so thankful to all the members in the Wagenseil lab for all their help this summer and inviting me, welcoming me into the lab, as well as all the people with CEMB and making this a really great summer.
What's the second question?
Okay. What is cannulated? Cannulated is there's two metal pieces on the myograph, and basically you stick the vessel like a sock onto it and then tie it with sutures on each end under a microscope sitting there.
I'm now convinced that I don't that if I go out and eat a bunch of it's gonna be just fine by aorta. What would change these stretch?
Sure. So when you have something like an aneurysm, the elastic fibers that I pointed out in this middle results column, they actually fragment apart. And part of what the research is doing in our lab is trying to figure out why that happens. And we know that about 20% are genetic, caused by genetic mutations. However, the other 80% are caused by things we're not sure yet, whether it's other genetic mutations, and they're connected with risk factors such as unhealthy diet or smoking. So things that would change the transport is if we have these fragmented elastic fibers. And we're trying to figure out exactly what's going on when it is having a greater transport, what's interacting less with the wall, I guess, and why that barrier is protected or not protected. There you go.
Questions?
She mentioned she had to step out and then she'd be back. She had something to take care of.
In that case, allow me to be there. Let's do a round of applause. And then, I am the last person in the room who should be walking. As the oldest person in the room, I'm the last person who should be touching the computer as we try to get to the next talk here. And look at this. Oh, no, back to the first talk. Is this the right one? You can give, by going second, you get to choose any of the remaining 4 talks to give. Is it? Oops. No. So this is a very short presentation. So actually, it's very interesting to look at what happens to plants when they're grown in the dark. Very interesting things happen. You're not Siena, usually. How do I make this go away? Oh, that's a good idea. So why did it go to that one before? All right, let's now. And so now somewhere in here there is a sorry. Oh, I think I'm going to try this one here. I'm old enough to remember these things. There it is. And now I'm pleased to welcome to the podium, all the way from Washington University, from all the way in St. Louis, and from all the way in the Huebsch lab on the third floor, third floor of this building, no less, Rapalino.
Hi, everyone. My name is Rapalino. As Dr. Genin said, I'm from WashU, where I'm a rising junior in the Biomedical engineering department. So this summer, I worked in the Huebsch lab, and the Huebsch lab in general works on tissue engineering, specifically making cardiac muscle in a dish. So using iPSC-derived cardiomyocytes, or reduced pluripotent stem cell-derived cardiomyocytes. So these cardiomyocytes are a more popular tool in cardiac research and also drug development. But one particular issue that they happen to have with them is that they are functionally immature. What this means is that as they are grown in this dish, they resemble embryonic cardiomyocytes instead of mature ones. And this has implications on their electrical conductivity, on their cell structure, and on how they beat. And so this is kind of a problem that we're looking to solve. And so one particular avenue of investigation is on the stretch that cardiomyocytes experience in the heart. And so we want to study how this stretch that they experience affects their maturity as they grow. So there are devices available commercially that can apply stretch to cells, but these devices tend to be really expensive, use proprietary materials, and generally don't really apply the stretch and type of mechanism that we're looking for. So I went about creating a low-cost, customized, I can get rid of that. Sorry. How can I move the Thank you. Yeah, so I'm creating a low-cost customized system for executing cell stretch. So this will hopefully allow us to image cardiomyocytes having been stretched and kind of see the impact that has on their maturity and other various parts of the cardiomyocyte. So to do this, I made a device. This device is essentially a small chamber made of polydimethylsiloxane, or PDMS. And this chamber is then screwed onto a vise. And as the vise is screwed and expands, the chamber is also stretched. And so cells that are in this chamber will be stretched as well. So to create this chamber, first I used Autodesk Inventor, which is a CAD software to design it. And then after printing it out with the 3D printer, I would coat this resin positive molds in agar to make a negative mold of resin, and then use that negative mold and pour PDMS into that negative mold to create the resulting chamber. This method was what was kind of originally made by someone else in the lab. And so after this chamber was cured, I would seed cells in the chamber. So far, we've used CT12 myoblasts. In the future, we hope to use cardiomyocytes as well. And then after the image, we would apply stretch after fixing the cells, and then we would observe the results. So while designing this device, I had to consider a few things to kind of improve the efficiency and make it so that it was superior to ones that are already on the market. So firstly, when making PDMS, it requires two parts, a crosslinker and a base. And typically, we use 10 parts base to 1 part crosslinker. And this is typically fine in PDMS, but in our case, we wanted to kind of maximize the amount of strain this can experience and minimize the elastic modulus. So to do so, I did material testing using an strong machine with several different ratios of the PDMS. So as you can see, there's the 1 to 10 all the way to 1 to 10, 1 to 12, 1 to 14, 1 to 15, 1 to 8. And we did initial testing for all those ratios using specimens. And then I determined that the ones that had the lowest modulus, in other words, that can achieve the greatest strain with the least amount of stress, were the 1 to 18, 1 to 22, 1 to 26 ratio. So given that, I then made the actual chambers out of those instead of using, I guess, controlled specimens. And again, stretch them until failure and figure out the ultimate strain that each one can achieve. And the one that we decided to go with was 1:18. So after determining this PDMS ratio, next I wanted to figure out where was the point of greatest failure on the chamber. Where was it most likely to fail? And so I ran an automatic simulation of the areas of maximal stress, and we determined that on these walls, on the edges of this chamber, that had experienced the greatest amount of stress. So preliminary kind of counteracting that thing, that stress was we filleted the edges of the negative to kind of make it so that the corners were at least less vulnerable to this stress. And so far it's been working pretty well. And we've kind of seen a consistent failure at that point. So after the material testing, next I wanted to make sure that this device was compatible with cells. So in other words, that if we didn't need any sort of coating or other mechanism to grow cells on this device, then it could just be done on PDMS. To do so, I seeded CTC-cell myoblasts that express the H2B fusion protein. And this allows us to image it with both the bright field filters and the H2B anti filters, or TRIC-C. As you can see, a little bit faint on this, but as each day 2, day 3, and day 4, there's a noticeable increase in the cells, and the proliferation of these cells on this chamber. So, and as we've started preliminary work on looking at the cell adhesion to the chamber, but haven't really had conclusive enough results to that yet. Quick review, we're making a chamber to observe the stretch in iPSC-derived cardiomyocytes. Material testing told us that I guess not best, but ratio that we decided to go with was the 18:1 based across on ratio for PDMS. We seeded myoblasts on these chambers to ensure that the chambers were biocompatible, and we saw noticeable proliferation. And lastly, this overall design costs about $15, whereas similar commercial devices are upwards of $1,000 or more. So future work, this chamber is made for cardiomyocytes. So one big step you have to do is grow cardiomyocytes on these chambers. Generally, cardiomyocytes tend to be more fragile than myoblasts, and so that would be kind of a next step to see that whether these chambers can support cardiomyocytes as well. And as part of that, cardiomyocytes need to be grown in a structured way, so we can kind of see that they're talking about adhesion. And part of that is we have to put on a stencil on these chambers and make sure that when the chamber is stretched, there's no delamination of the stencil. Lastly, as you can see on these pictures, the vise we use is a commercial one, and it's kind of big and bulky. It makes it difficult to image with the chamber on the vise. And so one possible next step would be to make a new vise or look for a different commercial one that's a little bit more streamlined and we can image while it's attached to this vise. So of course, before I end off, I want to just thank the Huebsch lab, specifically Dr. Huebsch and Arian, both of whom have been a tremendous help for me this whole summer, and have both been mentors to me this summer and this past semester. Thank you. Questions? Yeah, so the 1 to 10, if you notice, sorry to interrupt you there, but has the largest standard deviation of all the results. And so at the beginning, there was some initial hurdles with kind of making the device and getting more practice at it. And so a lot of those initial results would go to 10. And then as I got better at it, the chambers were more intact and kind of more hardy. And so the results of the 1 to 10 are kind of our biggest like mystery, as it were, as to why it got such a high result when the rest of the kind of trends in a similar direction. Yeah.
But then or is there something else going on?
So we think that it was due to the large innovation. We think that it was something just a matter of like where the first materials made, and that that trend is not generally seen with the rest of the ratios. And so we kind of said that was not like a particularly significant result, as far as like it has some sort of other property that we didn't think about beforehand.
Oh, yes, you can have a problem. We have such a pushover of a session moderator. Your analysis that shows some deviation.
Not in particular, as far as like relative to the stress or anything like that. There was some heterogeneity in how the cells were spaced out. It wasn't exactly an ideal solution, but for the most part, this was like the general trends that were observed as far as the cell growth.
I was going to ask.
Not in particular. I think the biocompatibility part was not so much of it as the material, but more for just whether PMS grew at all. So like whether we use 1 to 10 or 1 to 18, that part was kind of separate from the biocompatibility. Biocompatibility was just to show that like PMS could support cells and could grow them as on its own.
I'm pleased to introduce Patricia.
Thank you so much. All right. Okay. Our next speaker is not Melanie, is Nguyen.
Hi, everyone. My name is Nguyen. I'm a rising senior from LSU. And for the summer, I had the opportunity to work in Dr. Amit Pathak's cellular mechanobiology lab, where we studied the different effects of collagen 1 and 4 on collective cell migration. So the types of cells used for this study were epithelial cells. An example of that would be your skin, as they are made of millions of epithelial cells that are tightly packed together. So epithelial cells have a special orientation called the apical basal polarity, where it plays a role in the cell's form and function. And most importantly, it keeps the cell laterally attached to the basement membrane. And so the basement membrane is a specialized form of the extracellular matrix. It's comprised of mostly collagen 4, and it protects the tissue and gives it for its structure. So in the figure 1 right here, which is a schematic model of cancer leading to metastasis, the purple squares are normal, healthy epithelial cells, while the disfigured blue shapes are the cancer cells. So these cancer cells are epithelial cells who have lost their polarity. And as a result of that, they have reduced adhesiveness and increased migratory behaviors. This could lead to the basement membrane degradation and allows cancer cells to invade into surrounding stroma, which are comprised of mostly collagen 1 fibers. And the different phenotypes are expressed by collectively migrating cells in response to shifts in local environment. So from collagen 4 in the basement membrane to collagen 1 in connective tissue. So we want to see the characteristics of collectively migrating cells as they migrate through different substrates. And we did that through different matrix stiffnesses. And to do that, we microfabricated hydrogels of different stiffnesses with specified ratios of acrylamide, bisacrylamide, and ultra pure water. The ratios are listed here, but in summary, for our soft substrate, we used a stiffness of 0.5 kPa, while for stiff, we used a stiffness of 50 kPa. And in figure 2 is a schematic of the gel fabrication and cell seeding procedure. So first we had to activate the glass surface. And to do that, we did it through plasma cleaning and adding Wagenseil. This would make the polyacrylamide gel adhere more to the glass surface. Next, we polymerized polyacrylamide by adding timid and we added timid and ammonium polysulfate. After that, we activated the polyacrylamide surface through sulfa-sampa and Huebsch buffer, and then we cross-linked it to the polyacrylamide surface using a UV crosslinker. Then we coated the gels with the collagen 1 and collagen 4, and then we stored it in 4 degrees Celsius overnight. And then finally, we seeded the cells onto the gels, where collective cell migration was imaged using time-lapse microscopy, tracked using ImageJ with a TrackMate plugin, and quantified using Python. So what did we see for the results? So in figure 3, we have a collection of bright field images of growing epithelial cell monolayers with a quiver plot overlay showing migration speeds. For the speed scale, cells that were traveling the fastest at 40 microns per hour are dark red, while cells that were not moving at all are dark blue at 0 microns per hour. And we can see the 4 conditions on the left with collagen type 1 and collagen type 4 on soft and stiff substrate. And from these images, we concluded that cells on stiff substrate migrated faster than cells on soft substrate, regardless of ECM protein. And you can see that there are more red dots showing that these cells migrated faster. And in the next figure, figure 4, there are 6 graphs, each with a specific parameter that measured collective migration. So for the first row, there are 3 graphs plotted over time, over a 24-hour time span. And then for the second row, it is a bar graph that averages of these specific parameters. So now I'm going to go and explain what each of these graphs are. For the first one is MSD, the mean squared displacement, which measures the change in the cell's position with respect to a reference position. So in other words, it measures how far did the cells move. So in the graph, we can see that collagen 4 stiff moved the farthest, had the highest MSD values, while collagen 4 soft had the lowest MSD values. And then below it is a bar graph of speed, which, as mentioned before, it reinforces the claim that cells moved faster on stiff substrate compared to soft. And then next we have persistence and angular persistence. So persistence measures how linear the cells travel in substrate. So a persistence value closer to 1 would mean that the cells traveled relatively in a straight line from point A to point B, while a persistence value closer to 0 would mean that the cells chose like an unoptimal route as they traveled more distance to reach to the end position. And angular persistence is just the angle measurement of that. So the angle measurement of the cell's deviation from the end position. And from here, we concluded that cells on collagen 1 were more persistent on soft than stiff substrate, and cells on both collagen 1 and 4 soft showed initially greater persistence while gradually decreasing to similar levels as cells on stiff ECMs. And for the last column, we have order parameter and directionality. So order parameter measures how organized the collective group of cells are as they collectively migrate. So an order parameter closest to the value of 1 would mean that they were very organized and they traveled more collectively. And an order parameter value closer to 0 would mean that they would travel more chaotically and less organized. And directionality follows the same concept, except this measures each individual cell's ability to maintain direction. And here we concluded that, similar to the persistence, that cells on collagen 1 soft were had greater directionality. And then to the right, we have immunostained images, collagen 1 and collagen 4 stiff and soft. So we're missing collagen 4 soft, and I just wanted to note that cells showed resistance in monolayer formation. This could be due to a coding issue, or the cells just don't adhere as easily to collagen 4 softs. And this could be seen in figure 3 actually, as so all of these images, these cells, you can see the clear leading edges on one side, while on collagen 4 softs, you can see the whole monolayer in one frame. So it was very small and very difficult to grow the monolayer. So here we stained the proteins that are inside each of the cells that are involved in collective cell migration. So for green, we stained the nucleus and E-cadherin. And E-cadherin is involved in cell-to-cell adhesion, where it provides connection for each cell and allow them to migrate in sheet-like movements. And then for yellow, we stained paxillin, which is involved in the cell ECM adhesion. They are in focal adhesions, which are part of a macromolecule structure that provides mechanical links to the the cell's cytoskeleton and the ECM. And then finally, we stained for F-actin, which is a linear polymer of proteins that are a part of the cytoskeleton and contract and generate tension needed for cell migration. And some conclusions that we reached were that monolayer formation appears to be more efficient on collagen 1 coated substrates. In addition, the stiffness of the substrate may play a role in minimizing the protein effects, as we were able to readily form monolayers on collagen 4 stiffs, but not on softs. Cell migration were quicker but less persistent and organized on stiff ECMs. This led to a tangential migration at the leading edge on stiff ECMs, and this could be explained due to the quick monolayer expansion that the cells ran out of space on the gel. And for future work, we could incorporate a larger variety of ECM protein compositions in matrix coatings, such as laminin, matrigel, and fibronectin. We could do increased inflorescence staining on intracellular proteins to reveal subcellular mechanisms responsible for ECM protein sensitivity. And then finally, we could do traction force microscopy to determine effects of ECM protein on collective cell deformations and force generation. And for acknowledgements, I would like to thank all the members in the Pathak lab and Dr. Pathak himself. I would like to express my gratitude, especially to Chris and Hannah, for being great mentors and guiding me and helping me throughout the project. Thank you guys. I would also like to thank Dr. Van Horn from Bayer for his guidance and support throughout the summer. And I would like to thank Professor Widder and Ms. Nguyen for their support throughout the whole REU experience. Thank you. Yes. Yes, I thought about that too, but then so we did this on a 2D substrate, so we've decided that, or not decided, but like so it wouldn't have much of an effect on the cells migration as they traveled. But yeah, that was a thought. Yes. Yeah. Yeah.
But then,
So the time is in hours, over 24 hours. Yeah.
so after you go home on Friday and come back on Monday,
Yeah.
then the magnitude difference. That's what guys doing.
So we suspect, so on stiff substrates, the cells are able to migrate faster because of focal adhesions are able to like mature more and cells are able to migrate faster. So we think that because the cells were traveling faster, they were unable to migrate collectively or more organized. It was just chaos because they were going so fast. They weren't able to like reorient themselves in a organized manner. So I think it's because of the stiffness of the substrate that allowed the cells to just.
Then
Yes, I think that's so like cells on the outer edge are probably tend to move more, while cells in the center were like in place, they didn't move around as much. So it could have, skewed measurements of in the speed and in the MSD. Thank you.
I guess we should have asked if there's any questions from the remote attendees, but if there are, please do speak up. All right, but this is the time when we're supposed to take a break. So let's do that.
we value your circulation.
Please do circulate.
This is not the head out to me. I'm so sorry. Can you get a picture if you have together? The poster in the background, you know this is going to end the program exit. I hope this is only the beginning of the
It started to hurt.
She was smiling.
It was the
I'll put it on the perfect. Thank you.
It's not smiling all the time.
Okay, we're going to resume our presentations now. We have two more students to present. Next will be Kenadie Doty from the Boston Lab.
Hello, I am Kenadie Doty. I am going to be a senior this year at Buena Vista University in Storm Lake, Iowa. And this summer I had the pleasure of working in the Boston Lab with Morgan Lee as my bench mentor. So this summer, what I was really working with and learning about,
Sorry, I forgot to share.
you're okay. I'm gonna wait a second because otherwise I'm gonna be distracted, if that's okay.
I'm so distracted.
It's okay to be distracted.
Should I start over?
No,
Okay.
it's okay.
I get you.
Oh, yeah, as you wish, as you wish. Oh, shoot.
Oh, there we
Okay,
go.
so-
You get rid of that. Do that.
That was Melanie, I think. Perfect. Okay, as I was saying, this summer I spent my time learning about and working with cellulose nanocrystals. Cellulose nanocrystals are derived from cellulose biomass. And essentially what it is, cellulose is made out of a glucose chain it's a polymer of glucose. And if you look at it closer, there is some disordered regions and then some highly crystalline regions. And so if we get rid of all of those disordered regions, we're left with that highly structured material, which are called cellulose nanocrystals. I'm going to refer to those as CNCs throughout the presentation. So as I said, they're highly crystalline and they're very small. They actually are they're a nanoparticle. They are 1/1000th times smaller than the width of the human hair. So they're very, very, very small. Because they're so small, they have a very large surface area in comparison to their volume. And so because of this, it's really like available to change the surface chemistry of these cellulose nanocrystals. And so what I did this summer was I was changing the surface chemistry of these cellulose nanocrystals with benzophenone. So benzophenone is a chemical that is actually used in sunscreen. So it's something that you all should be using every day. But it's a photoreactive material or chemical that when exposed to ultraviolet light, it actually forms covalent cross-link bonds with the cellulose nanocrystals. So the covalent bonds are bonds that are a lot stronger, forces that are holding things together a lot stronger than the regular intermolecular forces that are generally in, or that, I mean, they still are in cellulose nanocrystal, but the addition of the covalent bonds really strengthens the properties of the cellulose nanocrystal. So I was functionalizing the cellulose nanocrystal with benzophenone and creating these covalent bonds. So what we can do with these benzophenone functionalized cellulose nanocrystals is put them into soy protein nanocomposites, or that's what I did this summer, was put them into soy protein nanocomposites. And soy protein isolate is just a byproduct of the soy oil industry. And by itself, nanocomposites made of soy protein isolate aren't very strong. They have not very strong mechanical properties, and they are very, they're not very water resistant. So they attract a lot of water. So if I reinforce these soy protein isolate nanocomposites with the cellulose nanocrystal, it will increase the properties, the mechanical properties of the soy protein isolate nanocomposite films, and it will make it so the soy protein nanocomposites can be used for more things, such as like it can be used in tissue engineering and optics, and there's a bunch of industrial uses. And then, like I said, the cellulose nanocrystals can be used as a reinforcement material just because they are so strong. Another big application that I'm interested in and that I was looking at specifically with the soy protein isolate infused with the cellulose nanocrystals is biodegradable food packaging. So that's another thing about cellulose nanocrystals, is that it's coming from a readily available source that's renewable and very easy to get. And so that's why it's of interest of us to look into. So my goal this summer was to quantify the effects of UV initiated cross-links on benzophenone soy protein isolate nanocomposites. So to do this, I've never used this one. So to do this, I started by isolating the cellulose nanocrystal. So this is a molecule of the cellulose nanocrystal. So I did this by acid hydrolysis. So I used 64% weight sulfuric acid to hydrolyze the disordered regions of the cellulose and leave us with the highly crystalline structure. After that, I oxidized the cellulose, so that put carboxyl groups on the surface of these nanocrystals, which made it more reactive. So I would be able to put the benzophenone onto the cellulose nanocrystals. So after I oxidized them and made sure there was a surface coverage of carboxyl groups, I coupled benzophenone onto the surface of these cellulose nanocrystals. And so after this, I incorporated them into the soy protein isolate films. And I just used 10% for all of my films here. And with this, I was able to test the mechanical properties and do some water absorption testing to see how the cross-links that I was going to be creating are affected. So the last step is to do the actual ultraviolet activation of the benzophenone. And then some things I did to characterize my products throughout was I used conductiometric titration, dynamic light scattering, Fourier transform, infrared spectroscopy, and then I also did mechanical testing and water absorption testing. So the first thing that I had to do during this process was, in between each step of the original CNCs to the oxidized CNCs to the benzophenone coupled CNCs, was I had to characterize them to make sure that they were actually what I thought they were. So dynamic light scattering can be used, or I use dynamic light scattering, which can be used to estimate the size of the nanoparticles I'm using. And it also gives us the zeta potential. And so how this instrument actually works is it shines a light through the solution with the cellulose nanocrystals, and it reads the change in light intensity as a function of time of the particles moving. And so based off of that, it's able to determine an estimated size. And my cellulose nanocrystals were in between 100 to 300 nanometers long. And then in figure 2 right here, you can see the zeta potential voltages of the cellulose nanocrystals. So the specific number that the zeta potential isn't super important, but the relative magnitude of the zeta potential is. So the zeta potential pretty much tells us about the surface charge of the cellulose nanocrystals. So the higher magnitude value of the zeta potential, the more, so like, for example, the tempo, see here in red right here, they are the furthest negative, which indicates that they have the biggest negative charge on the surface of the cellulose nanocrystals. And the negative charges are going to repel against each other and create a more evenly dispersed, stable solution. So it's important for us to characterize this, because if we are getting a solution that doesn't have a very high zeta potential or doesn't have a large magnitude, the dispersion isn't going to be very well in anything, let alone the soy protein isolate solution that I'm making. So it'll aggregate together and not be dispersed evenly, which will 100% affect the way that the nanocomposites perform. So the next thing that I did was I used red spectroscopy, which you can see here in figure 3. And so my goal in using the FTIR analysis was to compare the CNCs to the tempo CNCs originally. And right here you can see that, so this top blue line is the cellulose nanocrystals. And when you're comparing it to this red line, which is the tempo cellulose nanocrystals, you can see they're formed, which are indicative of the carbon to oxygen bond of the carboxyl groups. And so that just further confirms to me that cellulose nanocrystals really were oxidized. So then if we look at the BP or benzophenone CNCs here in green, we can see that this area right here compared to this peak area changes. And it's a little bit difficult to see because our BP CNCs still have some carboxyl groups, but the change at about 1600 and indicate NH bond, which forms when the benzophenone is bonded to the cellulose nanocrystal. You can also see that there is some, over on this side of the spectra, there is some carbon-carbon interactions that indicate, or indicative of the aromatic rings on benzophenone. And so this just confirms that my products are what I am saying they are. So then the last thing I did to characterize the actual CNCs before putting it into the soy protein films was conductiometric titration. So I titrated my tempo CNCs and my BP CNCs. And what this allowed me to do was just determine the amount of surface carboxyl groups were on the CNCs. Here you can see in red the tempo CNCs before I added the benzophenone and then after. And there was actually a 70%, almost a 70% decrease in carboxyl group content. And so that's indicative that the benzophenone is linked at all of those areas. So once I added the benzophenone into the soy protein isolate films, I also loaded tempo CNCs into the films, because to be able to make a direct comparison of what the crosslinks, what the effects of the crosslinks from benzophenone are doing, we have to ensure that the tempo CNCs in the soy protein isolate film aren't creating stronger interactions. And so that's why it's important for us to make tempo CNC films and BP CNC films. I did mechanical testing on all of these films. And so mechanical testing, in figure 5 right here, you can see an example of one of the stress strain curves of the mechanical test that I did. So essentially what the mechanical test did was take the end of each, or take one end of each, it would take the ends of one film and it would pull it apart and measure the distance that it was being pulled apart and the amount of stress it would, or the load that was being put on the films. And so I was able to get I got 3 different values from this. And so I measured the ultimate tensile strength property, which is just essentially the mass or the max stress that the cellulose nanocomposite films could go through. And you can see that right here. And in all 3 of the mechanical properties that I measured, there was a significant difference between the UV activated BP CNC films, which is indicative of the covalent bonds that are forming from UV activation. And you can also see that there's not much of a difference between these tempo films right here before UV activation and after UV activation, which further confirms that there's nothing going on in the soy protein matrix that's creating these stronger bonds. So the toughness was measured right here, and I actually had a highly significant result here. So the toughness value increased by almost double, or by a little bit over double the amount. And toughness is just the energy the material can withstand without breaking. So that's just going to be the area under this stress strain curve. And then I also got Young's modulus values. So that's going to be the linear region of it's measuring stiffness based on the linear region of stress over strain right here until the material deforms. And so all 3 of these results showed that the UV cross-linking aided, or it's indicative that there's covalent bonds because the chemical properties of the materials increased so much. So then the last test that I did on my films was water absorption testing. And so here we can see my water absorption test results. I initially I dried out my films and I got a dry weight, and then I put them in a room with 98% humidity and re-weighed the films after. And here you can see that both tempo CNC films, UV and no UV, had a lot of water uptake. Their weight increased by almost double. But then the BP CNC films here, you can see absorbed less water. And that's because benzophenone has hydrophobic properties, which makes sense as my results show that they're less hydrophilic than the tempo CNC films. But then what's especially important is that the BP films with UV activation have absorbed even less water, which makes sense because the covalent bonds are are stronger attractions to each other, keeping the water from being absorbed into the films. So this further is indicative of the covalent bonds that are formed in the UV cross-linking. And that's specifically the water resistance property is of interest and important because, like I said earlier, the ability for these nanocomposites to be used as biodegradable food packaging, that's a property that we would like to have, is not something that's going to just soak up all the water in the air when it's holding food packaging. But I just wanted to thank the Foston Lab and all of the members in the Foston Lab, specifically Morgan Lee, my bench mentor, and Jerry Wang, who helped me with the mechanical testing. And then I'd also like to thank Dr. Marcus Foston for all of his assistance. And then the Center for Engineering Mechanobiology, Washington University, and the National Science Foundation. And I don't have any questions.
The question that I won't ask was that we get 2% humidity.
So the films that I made were dog bone shaped is what they're called. And so at the ends of the films, it's thicker. And then at the middle, which is the part that we really care about, it's thinner. And as it stretches, some of the films already have cracking or not cracking, little abrasions in it due to the way it dried. But the ones that didn't, as it pulled apart, it did get clearer or more translucent as it broke, and it would just, like, cracks would form and rip apart. It depended on the film, but ideally it wouldn't have been like a lot of different, or in general, it wouldn't have been just a lot of different cracks at different spots. It would just be in the one spot with the most stress of being applied.
Quick question. I'm just curious more about the water resistance properties of these. That they are not...
So they will absorb water. When I was finished with the films, I could see a visible difference between how much water would absorb. Like if I put it directly into water, the Tempo CNC SPI films got a lot bigger. And I mean, they didn't repel water, like it absorbed water still, but the amount that it expanded in the water was, the Tempo CNCs had a lot more expansion in the water than the BP CNCs did. And that's something that we could measure in the future.
So something that's a scenario where at the end of the day, what you've done here is that no problem. You can put protein and you're taking what stands between
I am not quite sure the health implications of So I know that soy protein isolate is completely, or from what we know, it's safe to consume, and it's in a lot of like protein powders that people use. I'm not sure about benzophenone though. I would have to look into that further.
So it's in sunscreen.
Yeah, it is in sunscreen.
Exposure is probably-
Yes.
Inject-
Yes, I'm just not sure about ingestion.
Also get exposed to too much.
I think so. I don't know.
So
Is there anything bad in there?
at least bad, I'm not quite sure the health, we might have to call Center for Poison Control about that one. But what's stopping or what's preventing the production of like the completely biodegradable food packaging out of this, or like why people would continue using synthetic materials, is that we're still learning about this, like these different protein nanocomposites that can be used to make the bags. Yeah. Okay.
How do you know how the water is? So the benzophenone is, like I said, it was more hydrophobic. So hydrophobic means not. But the tempo CNCs have carboxyl groups on the end, which they have more hydrophilic properties.
More
answer.
like intramolecular
Yes.
going on less
So,
water and like
so in just the regular BP CNC films, like these one, or not, these aren't films, just the regular BP CNC, there isn't any covalent bonding yet because I hadn't exposed it to the UV crosslinks. So it's just the fact that the benzophenone, which covers the surface of the cellulose nanocrystals, which is a lot of the cellulose nanocrystal, has more hydrophilic properties than the tempo CNC. Other questions?
And thanks, Kennedy. Our next presenter is Sienna Smith. I can get to her thing. There it is.
Hi, everyone. My name is Sienna Smith. I am from the University of Michigan, Dearborn, and I'm a rising junior in the biomedical engineering program there. This summer, I have done work in the Meyer Muscle Lab, which focuses on how muscle changes in response to applied forces. And so my specific project has been dealing with how a protein called follistatin, how it affects muscle regeneration and injury response, specifically when adult mice don't have that protein. So first, follistatin is an actin-binding glycoprotein that antagonizes myostatin, which inhibits muscle growth. And so there's been a lot of work done on the overexpression of myostatin, but less so on follistatin deficient mice because follistatin deficiency is neonatal lethal. So we wanted to find out what the importance is of follistatin in after muscle injury in the short term. And so we did this by using inducible follistatin knockout mice. And so the way that we did this was by crossing two mice to allow that make them susceptible to a drug called tamoxifen. And so that was the beginning of our experimental timeline. We started by treating each mouse for 5 days, followed by a 2-week washout period to allow for the follistatin protein to fall out of the genotype. And so we ended up with 8 follistatin knockout mice and 6 littermate controls, which I will refer to as wild type mice in the rest of the presentation. Following the tamoxifen treatment, we started on the force treatment, which was our testing portion of our experiment. So what this included was using a very specialized physiology machine, which you can see in the lower right. So each mouse was anesthetized and we shaved their legs so we were able to see better where we needed to place needle electrodes in order to stimulate the perineal nerve. And the reason we chose this nerve is because we wanted to cause contractions in the tibialis anterior muscle, which is like your shin muscle. And so the first day of contractions would include, so we start by taking pre-tests of isometric contractions. And so an isometric contraction is, we intended to have it as a non-injurious control so that the muscle was not extended while force was being produced, so that in theory the muscle would not undergo injury during this contraction. This is opposed to the other type of contraction that we use, which is eccentric contractions, in which the muscle is extended as force is produced, which actively injures the muscle. So before we did any type of exercise protocol, I took isometric contractions to see how much force was produced by the muscle on the first leg of each mouse. Then I carried out a exercise protocol where I subjected each mouse to 10 sets of 6 contractions followed by 50 seconds of rest. And so this ideally was supposed to be a challenging protocol, but also to allow the muscle enough time to recover in between each set. And so after each leg of each mouse went through this protocol, we also took post-test isometric readings in order to figure out, okay, how much force was lost as a result of this protocol. And for isometric contractions, ideally you would want to see a pretty consistent force value in the beginning and at the end, because that was supposed to be a non-injurious form of exercise. And then, so once we got those measurements on the first leg, we would switch to the second leg on the same mouse. And so this would be our eccentric contraction leg. So if you see on the image to your right, there is a force plate which you can either leave stationary or you can rotate it. And the reason that we want to rotate it is so that we can dorsiflex the foot as force is being produced in order to extend and therefore injure that muscle throughout the exercise protocol. And so we again took pre-force measurements before doing the exercise testing. And then we did the same technique, which with the 10 sets of 6 with 50 seconds of rest. But this time the muscle, every time there was a contraction, the force plate would move and move the foot with it. And so on day 1, after this was complete, we would allow the mouse to rest and give it some painkillers, and it would get 1 day of rest before we went back with all the mice and did them again on day 2. And so the protocol was the same on day 2, but the only thing that was different is that after we finished the eccentric contraction bout, we gave them an injection of an antibiotic called purimycin. And then we harvested the supraspinatus muscle as a control and both tibialis anterior muscles 1 hour after injection. So that was the procedure that we kind of followed. So now I want to go into kind of all of the data that we were able to collect. And the first group of that is mass data. And so we kind of expected sex to be a contributing factor. So that's why you can see in this graph that we have these 2 graphs are kind of grouped based on male and female. However, we found that not to be the case when we measured the exercised tibialis anterior muscles in this case. So these are legacy measurements from a previous cohort of mice, and you can see that overall the mass of the muscle is lower in the follistatin knockout mice. And it was important that we also measured the muscle mass of the supraspinatus because that was also our control for the cohort of mice that I worked on. And so what is similar between these two blue and red graphs is that both of those were non-exercise. So even though those muscles were not subjected to a contraction protocol, in general, the mass of the follistatin knockout muscles were lower than those of the wild type. And so that leads us to the orange and purple graph up at the top, which also shows the interaction between the muscles in muscles that were contracted. And so this was 2 weeks after follistatin knockout. And so you can still observe the same pattern where the follistatin knockout mice had a lower mass, although there was not a significant difference between the sex of the mouse. And so the next group of data that we were able to collect was from also a previous cohort of follistatin knockout mice. And so that was this fiber typing data that we have below. And so fiber typing is basically when you take a section of muscle perpendicular to where the muscle fibers run, and then you're able to stain it and then image it to look at what types of muscle fibers show up and how big they are. And so I was able to do fiber typing analysis on a previous group of muscles. And I found that although the fiber type area was not specific based on genotype, we did find that type 2B fibers, which are fast twitch fibers, and type 2A fibers, there were more 2B fibers in follistatin knockout mice and less 2A fibers than in wild type. And so this data is important because it's when doing muscle research, fiber typing is a very common technique. So it was very important for me to kind of learn how to do this, especially in a muscle focused research project. And so at the bottom of the first results column is our force data. And so I want to preface this by saying, obviously you guys can see how variable the data is and how large the standard deviation bars are. And that was because it was so hard to get the needles in the right placement every time. And even if you did find somewhere that, okay, it gave you a proper force reading, 5-10 contractions later, it might die and there might not be a reading that you can get. And so the data was so variable. But so each graph, so it shows first was the pre-tamoxifen, so pre-injection force output. And then each consecutive bar shows, okay, day 1 pre-testing, day 1 post-testing, day 2 pre-testing, day 2 post-testing. And so it's basically as the muscle gets contracted more and more, how does the force output respond? And so the top graph again is the isometrically contracted, so in theory non-injurious form of muscle exercise, whereas the bottom is the eccentric contraction where the ankle is dorsiflexed. So there weren't any significant differences between the days, but we were able to kind of identify that force recovery, at least partially, was still possible without the follistatin protein. And so this is important because we know that, okay, even if a mouse doesn't have that protein, some muscle healing is still possible to allow it to continue to produce force when it's contracted. And so I want to now look at those two graphs that you see on the bottom with the white background. These further kind of show the variability of the force outputs that we kind of had to collect. And so the first graph with, this first graph kind of shows what we might hope the kind of eccentric contraction output trend might be over the course of the protocol. And so you see like a small decrease in kind of the slope of the line, and then a small increase, and then another small decrease. And so this is kind of what you would expect, because once as you're doing the 6 contractions, okay, the muscle fatigues, but then when you allow it to rest, it kind of regains some of that force, and then it continues to decrease throughout the whole protocol as the muscle continues to fatigue. However, and then the top line is day 1, and then the bottom line is day 2. But however, for the one on, for this one, we see that that pattern is just does not appear. And so we can see at this point, there's a complete drop in force, which again, not necessarily because the muscle just stopped producing like torque. It was just because of the variability of the data collection, and it was really hard to get repeatable data. And so you can see that there isn't a clear pattern from day 1 to day 2, or even within each of the days themselves. And so it kind of just goes to show that like this was one mouse to the other. And so we had 14 different mice. And so the data that we got from each mouse was just, it was so variable. And so that's why you see such large error bars and such kind of big variation in the data that we were able to collect. And so if we jump back up to the histology section of our results, the multi-part image that you see starts with, so part A, this is a 10x image of H&E stained supraspinatus as a control. And so this supraspinatus muscle wasn't injured in any way. So you are able to see, not as well on the screen, but there are nice healthy muscle fibers that you can see. And this is contrasted by image B, which is this big section. And so this is an isometrically contracted muscle. And there's a little area right here that is outlined, and that is an area of necrosis. And so that's basically muscle cell death. And so that is, we ideally would have wanted to see none of that in isometrically contracted muscles, because we wouldn't have wanted to injure the muscle. But we can compare it to this image over here, which is image D, which is an eccentrically contracted muscle, where you see there's a red outline portion, which includes a much higher percentage of the muscle that was unable to recover post eccentric contraction bout. And so each of these embedded images, so this shows muscle fibers at various kind of points in the regeneration process, whereas this 10x image from this eccentrically contracted TA basically shows that completely degenerated muscle fibers that weren't able to recover after exercise. And so we were not able to see kind of a difference between the genotypes necessarily, but we were able to, if you look at the graph over here, we were able to see that the eccentric contractions did injure the muscle more than the isometric contractions, which was ideally what we wanted when we designed the experiment originally. And so the final part of our results data are Western blot results. And so we blotted for PAKT and AKT, mTOR and PMTOR, and then S6, PS6. We also did actin and puromycin. We weren't able to get any great bands for mTOR, and again, it kind of, we just weren't able to see that the proteins showed up enough. And then we use actin as a kind of protein control for like band intensity. And then we were able to kind of get decent, at least decent images for the AKT and P and S6 pathways. And so what these are is, if you jump back up to this, you can't see it very well, but it's basically just kind of downstream in like the kind of muscle regeneration pathway. And so right here is mTOR, this is AKT, and then S6 is further downstream. And so basically what we were doing with this is we were trying to see how follistatin, or the lack thereof, affected muscle regeneration. And so the Western results kind of trended toward an increased puromycin incorporation in the TAs that were exercised, but there wasn't a specific genotype difference. And so puromycin again is an antibiotic, and so it again looks further at how downstream muscle regeneration is affected by follistatin. But yeah, so we were able to conclude that follistatin is required for mass maintenance, because again, there was a drop in every time in the masses of the follistatin knockout muscles as opposed to the wild types. And we were able to generally conclude that follistatin is required for force recovery, because it's it's not required for forced recovery because in the wild type and follistatin knockout mice, about the same amount of recovery was possible. But unfortunately, because of the experimental variability, our kind of study was not like statistically powerful enough to determine any further smaller differences between the wild type and follistatin knockout genotypes. But because there has been so little research already done on follistatin knockout mice, the fact that we really weren't sure what the injury response would be is still important in terms of finding out what the role of follistatin is after injury recovery. And the study also was important in helping learn more about how muscle regeneration is driven. And an important clinical implication of this is that there is a possible connection between sarcopenia, which is the muscle loss and weakness that just happens to humans with age, and follistatin deficiency. And so there's speculation that follistatin is partially responsible because less circulates through the body as we get older. And this is supported by the mass data that we were able to collect because the muscle mass in follistatin knockout mice is lower than those in the wild type mice. And also there are less fast twitch fibers that we found in the fiber typing data, and that is also the case in muscles affected by sarcopenia. So finally, this study focused more on what or kind of if something would happen as opposed to how. So I think that focusing on that in the future would definitely be very insightful. And unfortunately, harvesting the muscle only 1 hour after eccentric contraction completion, it really limits kind of how much we're able to figure out about the long-term muscle recovery without follistatin. And so future work would include kind of allowing ourselves to kind of look at that aspect of muscle recovery and also trying to reduce the amount of damage that we did do with the needle electrodes. And that might include changing the injury model that we choose. Obviously, more precision will just come with practice as well. And I also think that increasing the number of mice in each group would definitely increase our statistical power. But yeah, I really just want to thank Dr. Gretchen Meyer and Jake and Karen and Kay, who are all sitting back here from the Meyer lab. I literally could not have done it without you guys. I also want to thank Nguyen and Professor Widder. You guys were the best. And thank you guys. I'm happy to answer any questions. The plate that you can see it sitting on, it's hooked up to a water heater, so it pumps warm water. So one, that's more for the mice safety while they're under anesthesia, as opposed to kind of a control in terms of like muscle function, but they were all at the same temperature on like the heated plate that's connected to the physiology machine. Mm-hmm. Yeah, so so the qPCR is from a previous cohort of mice that we did the same treatment on. So basically that is just proof that the treatment that we did do does end up knocking out the protein. Unfortunately, I didn't have enough time to run qPCR on my mice that I did, and so we were able to use that data. Okay, we are assuming, and they're also significant. There's also enough differences between the genotypes in other areas, and so considering that we have the qPCR data from the same mice in a different cohort, we're able to kind of assume that follistatin was knocked out because we used a treatment that was successful in the past. Mm-hmm. Nope. That would have been a good idea. Yeah. Yeah, for sure.
First question is that oh, she's here.
Yeah.
Let's answer that one because Gretchen's here.
Yeah. So the biggest thing that I saw that was really dramatic between the wild type and the false knockout mice were after we kind of put them in a little heated box with a little mat, like a little heating pad and stuff, and they would drag the eccentrically contracted leg for a while after they were done, as opposed to kind of the eccentrically contracted leg. And there was a lot of swelling as well after we put them through these kind of exercise protocols, as well as tissue damage from trying to get the electrode placements in the right spot. So there definitely was kind of a lot of like external kind of, like you said, soft tissue kind of observations that could have been made. And I think I'm just thinking about it now, that would have been kind of useful for me to write that down less in terms of, okay, what specific testing results did I get, but also kind of like almost intangible things like what you said, kind of what happened as a result of the protocol for each of the mice. Thank you. Oh, there's one more question. That's okay. Yeah. I'm lucky to go to a school where I could even be here. And so I think that this was so helpful that obviously in terms of like exercise and stuff, like it's all science. So like kind of, I don't know, kind of working out in a way that doesn't obviously do what we did to these poor mice and just kind of kill their muscles, and kind of just that balance. And that's kind of why this project was so interesting to me as an athlete, because it was very tangible with these mice and also deal with kind of exercise, which is something that I love to do anyways. So yeah. Thank you.
Okay, this concludes the presentation part of our event this morning. I'm going to stop the recording and unshare the Zoom. Thank you so much to all the people that have been with us throughout the morning remotely. We appreciate you being here, and we're just going to have some last gifts and closing remarks that will be more informal before we go to lunch. So again, thanks to everyone, and great work to all the students.